Hi, good morning, everyone. So I have to start by saying that E. coli smell terrible. In fact, their stink has plagued microbiologists for over a century. So this summer, we, MIT's 2006 iGEM team, decided to address this problem by engineering a strain of E. coli that are scented and perfuming, OD coli, if you will. I'd just like to briefly introduce the team. We are 5 undergraduates. My name is Vina. I'm a sophomore majoring in chemistry and physics. This is Andre. He's a junior majoring in biology and physics. Stephen is also a junior majoring in biological engineering. Kate's a sophomore majoring in biological engineering as well. And Bo is a sophomore majoring in electrical engineering and computer science. Unfortunately, he has laryngitis today and will not be able to present with us. But we also had 5 excellent graduate students and 2 faculty advisors, Drew Endy and Tom Knight, who are invaluable. So I mentioned that we engineered these scented bacteria. Well, what were our motivations for doing this project? You don't have to travel far to see, or rather smell, why our project is so incredibly relevant. Just across the parking lot in the bio building here at MIT, there are countless labs where you just walk in and all you smell is the reek of E. coli. Our project fixes this by making the lives of countless microbiologists that much better by perfuming their labs with mint and banana. Another reason that we chose for this project was the fact that in biological engineering, we have investigated visual reporting devices, but scent has not really been investigated as a useful reporter. So in our project, we sought to expand this field of synthetic biology. So I just want to go into some background about making a scent. To make a scent, we found that you need to have an enzyme. And what this enzyme has to do is convert something that does not smell into something that does smell. So we found 5 of these enzymes that do this task for 5 different scents. However, as we progressed through the summer, we had to eliminate a few due to problems in the lab. For example, the jasmine scent could not be isolated from the DNA, even though we tried to PCR it out, and same with the cinnamon, we just couldn't get the DNA for that. There's also this 3rd scent, which was advertised in the literature as being fresh and floral. However, in lab, we found that it actually smelled like cherry cough medicine, and no one really likes cherry cough medicine, so we just tabled that one right there. So we were left with these 2 scents, wintergreen and banana. Wintergreen is encoded by the enzyme BSMT from the plant C. brewery, while the banana enzyme was isolated from yeast. I'm just going to walk you through an overview of our design process. We started in a phase of initial planning, where we investigated, could we put a precursor into a cell and have it give off a smell? So this great thing's a cell. Can we put something in and get something out that smells good? Well, we thought we could, so we went ahead and designed devices that did this. So as you can see, this green box represents a wintergreen generating device, which takes in salicylic acid from outside the cell and produces the scented compound of methyl salicylate, which smells like mint. We created an analogous banana generating device, which takes in isoamyl alcohol from outside the cell and converts it into isoamyl acetate, which smells like bananas. So this is a great system. You put something in, you get out your wonderful scents. But we want to take it a step further, because as you can see, you have to add salicylic acid and isoamyl alcohol to get this cell to produce its scents. What we wanted to do was to take these molecules and bring them into the cell so that we didn't have to add anything to produce our scents. So we did this here. We created these devices, a salicylic acid generating device in the green hexagon and an isoamyl alcohol generating device in the yellow hexagon. So what these devices do is they create the precursors that go into the final smell generating devices to create your mint and banana scents. So at this point, we had a fully autonomous, self-sufficient system that would create mint and banana scents. That's great. As biological engineers, though, we wanted to take it a step further and regulate our system. So in the next phase, we introduced this mechanism of scent regulation. So we want to tell our E. coli when to smell like banana, when to smell like mint. So we did this based on a growth mechanism that Stephen will go into a little bit later. I mentioned that persistent stink of E. coli. Well, we wanted to get rid of that. That background stink, it's no good. So to eliminate it, we had to find out what it was. And we found that the stink was caused by a molecule known as indole. So we researched several different strains and knocked out indole. And, well, we transported our system into an E. coli that did not produce indole. So in the end, we had our system in these beautiful smelling E. coli that you can smell later on during the poster session if you haven't already. I'm going to pass it on to Andre now, who's going to talk about our devices and how they work.
So what Vina has just shown you is a very zoomed out picture of what we were trying to do this summer. You see these green and yellow boxes, green and yellow hexagons, but what is actually inside of these things? These are our individual devices, and that's what I want to tell you about. I'll begin with what we called our Wintergreen generating device. As Vina mentioned, only one enzyme is necessary to go from the precursor, salicylic acid, to methyl salicylate, which is the compound that smells like mint. But we had to construct a device that would produce this enzyme. And it included DNA parts, including a promoter, ribosome binding site, and terminator. And assembled in this particular way, whenever you put this device inside E. coli and added salicylic acid, you generated the mint smell, the methyl salicylate. As engineers, we wanted to quantify this system. Did we correctly construct our device, and is it working properly? And this is gas chromatograph data that actually shows that this particular device did indeed work. In E. coli, obviously, there's no methyl salicylate being produced. E. coli do not naturally smell like mint. But if you add the precursor molecule, salicylic acid, and you insert our device into E. coli, you do find a peak that is indicative of the molecular weight of methyl salicylate, proving that methyl salicylate is indeed being produced. But for our particular system, if we really do want to make labs across the world smell better, it's not sufficient to say that just methyl salicylate is being produced. We want to actually be able to smell it. And that was the most important part of testing this particular device. And that's what we did. We added the precursor, added our device, and we were able to smell mint. And if you got a chance outside for our smell test, you may have actually been able to do the same. Furthermore, as Vina said, we had an analogous system for producing the banana scent. Again, one enzyme was necessary. We assembled the necessary parts, and once that was done, we could therefore take in, if E. coli could take in isoamyl alcohol with this device in place, they would produce the banana scent. Again, after we designed this device, we constructed this device, we tested it, again using our noses. And I want to point out here that it was really important that we came up with an easily testable system. And you came out this weekend to listen to these presentations as a vacation, but you probably didn't think coming that you'd be doing an experiment while coming, and let alone someone else's experiment. But the way our system is designed, anyone can test it, and that's what we had set up outside. So the two devices I've described to you so far are only somewhat useful to us because you still have to add an outside molecule. So we wanted to take this a step further. We want E. coli to automatically produce the precursor molecules. So in particular, to generate methyl salicylate, the mint-smelling compound, if you begin with charismate, which is naturally occurring in E. coli, and you insert our salicylic acid generating device, which has 2 protein products. These products will metabolize charismate and give you salicylic acid. Salicylic acid enters the wintergreen generating device, giving you a mint smell. And essentially the same system for the banana smell. Leucine, as you know, is already present in E. coli. It enters our isoamyl alcohol generating device, being metabolized by these 2 protein products. And 3-methylbutanol is the actual first result, but E. coli will metabolize 3-methylbutanol to produce isoamyl alcohol. Isoamyl alcohol enters the banana generator device, producing a banana scent. And now I will hand it over to Stephen to talk about how we wanted to control the system.
Thanks, Andre. Well, so the goal of iGEM in our minds was not only to make a cool system, but also show some sort of regulation over that system. So we looked into many control mechanisms, such as light-sensitive promoters and heat shock promoters, but ended up deciding on doing something that has never been seen in the registry. So we decided to isolate a stationary phase promoter, which was only active once the cells reached a critical cell density. Our goal was to implement this promoter as a switch, which would express banana in stationary phase and wintergreen in exponential phase. The reason that we picked banana in stationary phase as opposed to exponential phase was because the banana smell, as you might have been able to see already, was much more potent than the wintergreen smell. So we thought that it might be able to overpower any residual wintergreen molecules that may be left over from exponential phase. So we researched several promoters, and we finally decided on OSMY, which is active in stationary phase and under high osmotic pressure conditions. So we hooked up the OSMY promoter to the banana smell generating device, so it would produce banana in stationary phase. And likewise, we connected the OSMY promoter to an inverter plus the mint structure to express mint in exponential phase. So you might say, oh, this seems like a great system, but how are you going to be able to test it? So the way in which we did that was we hooked up our OSMY promoter to our GFP protein generating device and measured fluorescence over the cell growth cycle. And this data, on the x-axis, you can see absorbance, which is proportional to cell density. And on the y-axis, you can see GFP signal, which is proportional to the GFP molecules produced in the cell culture. And as you can see, the red line represents the OSMY structure connected to GFP, and you can see a very clear increase in GFP production around 0.35 absorbance, which is indicative of stationary phase. Also, you can compare this with the gray curve, which is a relatively linear relation, which is the constitutive promoter connected to the GFP protein generator. And also as a negative control, we had the brown curve, which represents just cells with no fluorescence whatsoever. But we decided to take it a step further and connect the OSMY promoter to the inverter plus the GFP protein generator. And as you can see from the green curve, GFP production levels off at stationary phase. after rapid growth during exponential phase, indicating that our structure worked. So now I'm going to hand it over to Kate, who's going to show you how these control mechanisms fit into our overall system.
Thank you, Stephen. So now I'm hoping that you're all a bit more familiar with our 4 protein-generating devices, as well as our novel stationary phase switch mechanism. And so now I'm going to blow us back up into the big picture and recap just how, by assembling these specific components, we are able to successfully engineer and regulate our 2 different scent systems within the same E. coli cell. So as you can see in this first slide, our cell is in its exponential phase of growth, which means that due to our awesome Y inverter sequence here, methyl salicylate is in fact the only scent compound that can be produced. And if you'll stay with me as we fast forward a few hours in the cell growth cycle to its stationary phase, when it's reached a higher cell mass density, we can see that the awesome Y inverter will no longer control the transcriptional activity of the system, but the plain awesome Y promoter connected to our banana generating device will overtake the system control, and we will get only banana during our stationary phase. So we see that we have this regulated system capable of producing methyl salicylate during exponential phase, capable of producing banana isoamyl acetate during stationary phase. And I'd like to emphasize that each one of these devices we were able to design, construct, test, and prove that worked this summer. And we added it into the registry. So you might ask, what more could we do? Where could we go from here? Haven't we reached the end of this project? But we wanted to do even better. We wanted to improve our system even more, and we figured that the best way to do that would be to eliminate the natural sink compound still present in our E. coli. And we went back to the literature to do this and found that, again, indole was the molecule responsible for the bad odor. We found out that it was biosynthesized within the cell from the tryptophan. And gleaning from our experience looking into biosynthetic pathways this summer, as we designed our 4 biosynthetic protein generators, we figured and hoped that mutating the tryptophanase gene would yield us with indole-deficient phenotypes in our E. coli. So what we did is we obtained from the Yale Distribution Center a number of strains with various mutations, deletions, and insertions in the tryptophanase gene, grew those cultures and smelled them, and found our clear favorite, and then took that on to characterize it with a gas chromatograph again. So we have definitive data showing that, as you can see in our wild-type E. coli, the characteristic indole spike is our problem, and in our ideal O to E. coli chassis, there is no such spike. So, even though we're MIT students and have each 300 tests this week, we managed to bring our final system into this final chassis. We have yet to fully characterize this full system in this full chassis. However, as we've mentioned before, we have in fact characterized each part individually and various permutations of the scent subsystems in wild-type E. coli as well. And we are confident enough in our final system and final chassis that we were actually demoing components, pieces of that final system in our final chassis for you today. That was our indole deficient strain you were smelling. And this actually is the compilation of this morning's smell test results. I hope you were able to participate, and if not, we are going to continue that smell test this afternoon at the presentation demo, the presentation posters. And as you can see, about 90%, it looks like, we're able to correctly identify our banana culture. And Stinky had a pretty good success rate too, and pretty not bad on the mint either. So I'd say that's pretty good data. Congratulations, guys. Thanks for helping us out. And briefly, we'd just also like to mention the future applications we envision for our project. If we've already shown you that we could port our E. coli, or our system into a new strain of E. coli and eliminate stink in the lab, who's to say that we can't port our system into other bacteria and eliminate more stink elsewhere? If we were to port our system into biofilter or bioremediation bacteria, we could potentially make bacterial perfuming biofilters. And what about porting our system into the bacteria that are in your armpits, or bad breath bacteria as well? So, you know, bacterial deodorant, you never know, it's out there. And another exciting implementation that we envision is if we ported our system into common baker's yeast and started baking with that, we could design some new engineered scents and flavors in bread and beer. I mean, who hasn't wanted to try banana-less banana bread or minty fresh beer? So, anyways, in conclusion, I just want to sum up and say that we have designed, built, and tested a synthetic biological system over the course of one summer. We've engineered a novel synthetic scent system in E. coli, and we've, really importantly, regulated that via transcription-based control devices. We want to thank you all for attending our presentation, for supporting iGEM in general. It's going to be a great weekend, and we'd like to also acknowledge the generosity of Natalia Dudureva, Aaron Pechersky, Mary Berlin, Brian Cook, Cornelia Riemann, Dieter Haas, Peter Backer, and Herbert Schweizer and Pamela Silver, because without them and without their support, none of this would have been possible. And we'd like to open up for some questions. Thank you. Okay, we can call on you.
I was just wondering if you see any changes in growth rate when you change the smell from bad to good.
Do we put that slide on? So Jamie, I believe, from Brown, was actually wondering if we observed any different growth rates when we put our system into our cells. And Stephen is the expert on that, so I'll hand it over to him.
Yeah, we did witness a bit of a slowness in growth, but we also noticed that for like many constructs in general placed into the cells as compared with wild type cells. So. Oh.
That's a really good question. He was asking if producing our scent protein, our scent enzymes, will interfere with lab work if people are studying E. coli in the context of expressing their own experiments. It's a good point, and yes, I do believe that perhaps these enzymes would interfere with certain mechanisms. However, that's something that would be on a case-to-case basis, I feel, based upon what that individual researcher is studying in his cells or her cells. So, yeah, it's a good question. Sorry.
So when you took all this the group of genes and you put into this E. coli, did you need to like optimize the whole pathway for expressing this thing in E. coli? Just like you took the natural genes and they got expressed in E. coli?
Okay, so he was asking if we had to optimize codon optimization to express. Oh, yeah, optimize our whole expression, because that for E. coli expression. So one of these, for the Mint enzyme, it's already been expressed in E. coli, so that we knew would work. And the ATF1, which was the banana generating enzyme, is straight from yeast. However, that too has been expressed in E. coli. So, yeah, we did look into that.
Getting significant levels of sort of sticky recognition in strain A from your cell line, that had some of the highest background levels. Are there other things that you follow the second class of compounds that people pick up on?
So we did a good amount of research. Oh, so in the front row here, he was wondering if, since there was a little bit of background data for our strain A, which was our minty bacteria, if there was any other compounds that were needing to be knocked out to get a final clean chassis. And through the literature, it wasn't extremely specific. There could be another molecule called scatol, which is potentially involved. Yeah, that's lovely. Sorry. But so far, if we were to just, like the indole deficient are a significant step up from the stinky. So for now, we don't know of any other improvements to make. Okay, one more question. We'll go with the blue stripes.
Do you actually use these minties and bananas and stinkies for mini preps?
Yes, I mean, oh, so he was wondering if we used our mini preps from. We couldn't? Why? It's the same DNA. So he was wondering if we could use these minty and banana cells to grow up and use and then spin down and make mini preps out of, right?
So actually, if you're talking about transforming plasmids into competent cells, we used competent cell, top 10. We used just standard top 10 competent cells with no. Oh yeah, we can make these competent and transform other plasmids into them, for sure. Actually, just from using these cells, our whole 37-degree warm room smelled like a bakery or something, so it was great. Do we have time for one more?
I think I missed it, but did you show data that in the final construct you actually had IAA expressed in the stationary phase and Mint expressed only in the growth phase? From GC data showing this?
Okay, so that's what we're waiting on. Like, that's what we're doing right now, but we have shown that the systems work independently. But no, we didn't present data for the system in the same cell. That was just constructed like several days ago.
Thank you very much.